Welcome to part 2 of our Special Senses Notes, focusing in on gustation, or your sense of taste. So, when we think of taste, usually we think of taste buds, but maybe your perception of what a taste bud is isn't accurate. For a long time, when I thought of taste buds, I thought of these little bumps on your tongue, and I called those taste buds. But they're actually not taste buds. They're called papillae. Lingual papillae, to be correct. So, sense of taste is based on taste receptors, also known as gustatory epithelial cells, and they are found inside of taste buds. But again, the taste bud isn't those little bumps on the tongue. They are within, taste buds are found within 3 of the 4 types of lingual papillae. There's valate papillae, foliate papillae, fungiform papillae, and filiform. The 3 highlighted ones or bolded ones are ones that you do have taste buds in. The filiform ones do not have taste buds in them. And why would the filiform ones be there if they don't have taste buds? It's thought that the filiform ones actually help offer friction to food and getting it moved around in your mouth as you're chewing. So kind of controlling the food. If your tongue was a smooth, slippery surface, that wouldn't be good for controlling where the food goes. Your book does give some diagram views of the different types of papillae if you want to compare them, but we won't be tested on that. So if we were to look inside a taste bud, so here is a papillae, and then inside the papillae, these little bumps, within there would be these little receptor pores that would go into a taste bud. And then within the taste bud, there are receptors. So here is a taste pore where little bits of saliva containing dissolved food particles would go into and interact with your taste receptors. And so this is the taste bud. These are the taste receptor cells. You'll notice they are connected to sensory neurons, taking that information in. And then you have some other cells next to the taste receptors and you have to have a way to replace those. So again, within a taste bud, they are found within those little papillae, those little bumps. Within each taste bud, you might have 40 to 100 different taste receptors, so these kind of cells. And on the ends of these cells are what are called microvilli, or taste hairs, and they extend into the taste pore. So the little bit of saliva containing your food particles goes into the taste pore, interacting with the little microvilli at the tips of these taste receptor cells. The microvilli are going to help increase the surface area too, and they're going to be lined with receptor proteins. And so our taste receptors are actually next door to transitional cells, which are going to replace the taste receptors as they die. So they only live about 10 days. So you're going to have some basal cells that are going to be growing and maturing into new taste receptors. This would be a histological look, cross-section of a taste bud under a microscope of a real stained taste bud. And when we think of taste, you may have learned about the different tastes that our tongue can perceive. It's worth noting a couple things. The perception of our taste is tied directly to olfaction, our olfactory senses. In fact, you're able to distinguish taste with thousands of times more sensitivity when your olfactory receptors are assisting. So if you plug your nose and try to taste something, it's not going to be nearly as effective for distinguishing taste. And we've learned probably in elementary school, primary tastes are sweet, salty, sour, and bitter. And you may have learned that some regions of the tongue show extra sensitivity to one of these tastes, but that's not the whole story. This tongue map that you may have learned about in elementary school could be wrong. What is known now that is all the taste buds that have taste receptors are able to provide sensation for all the primary tastes: bitter, sour, sweet, and salty. So all taste buds are able to respond to those things. So you may have learned in elementary school, but actually the tongue map is not very accurate. In fact, I've linked on the slide here to a news story that talks about why this is no longer accurate and why we got a misconception early on from back in research in 1901. So because parts of the tongue are more sensitive to certain tastes, it got mapped out as this is the place that's responsible for that taste, even though that is not accurate. And in fact, some of the tastes we are more sensitive to are sour. So we're 1,000 times more sensitive to sour tastes than sweet and salty. Sweet and salty don't pose much of a risk, but potentially sour associated with acid could be harmful to mucous membranes lining the pharynx and throat. You don't want that. So we are tuned into that taste pretty strongly. Even more strongly, bitter, we are 100 times more sensitive than even the sour taste to perceive bitter. And that's because a lot of plant toxins are bitter. And so a lot of biological toxins are bitter. And so we tune into that for life-saving. So if you eat a berry and it's bitter, you might think twice, because you don't want to have toxin. And those are just plant defenses. Two other tastes that aren't as well known and aren't mapped on the slide, but I found interesting. Umami is a Japanese word that means, I think, savory. And it's associated to receptors sensitive to the amino acid glutamate. And so there's that taste perception that we are able to determine. And then water receptors. There actually are water receptors in the back of the tongue. I think it's the velate papillae taste buds area and the pharynx. There are water receptors that, again, send signals right to your hypothalamus. So that is part 2 of Special Senses Notes on gustation and our sense of taste.